My name is Tyler Nguyen, and welcome to my presentation about NMR. Currently, researchers are looking into possible technologies that are able to conduct real-time monitoring of compounds in living organisms. Particularly, scientists have been able to isolate cells from many different tissues, including from mice and even humans, and study them using a technology called NMR. The problem, though, is that these can take ages. The particular research that I want to focus on today is a study that looks at the monitoring of a helpful antioxidant named glutathione, which helps avoid your cells from dying due to things like pollution and eating too much turkey and ham for Thanksgiving. They investigated the issue of time and attempted to prove that NMR can be viable for this monitoring on the scale of minutes compared to hours. So why is this so important? Your body is made of literally countless pathways of complex compounds and enzymes, which help to keep you alive and active. These kinds of monitoring techniques that I will describe today have countless clinical applications and could even save your life one day. For example, the pathway that the researchers studied in this paper focused on an antioxidant, and if your body isn't able to replenish them, then it's very possible, in fact probable, to get things like cancer, Alzheimer's, and heart disease very easily. This technology could help diagnose a deficiency with a lab test, just how urine is tested at the clinic. Here's the pathway that we will investigate today. The most important thing to get from this is three things. Cysteine is the building block for GSH, the antioxidant from earlier. Then in the cell, GSH can turn into GSBIM, which is an alternate form of GSH that can get taken out of the cell. When stress occurs, GSH helps out, turning into GSSG. This creates a need for more GSH, which can be created using more cysteine. The way that GSH turns into GSBIM is through a compound called MBBR. I will also describe NMR, which is a technology being used to monitor the alive and thriving cells. First, an alternating magnetic field is generated in the machine. This causes carbons within the molecules to start spinning, which then generate a signal that can be picked up by the instrument. There's a ton of noise with this process though, so the machine will take thousands of snapshots and combine them, filtering the random noise out of the scan. So here's how the experiment went. They first created something called carbon-13 labeled GSH. Then they saw how GSH reacted with MBBR. They then plotted the intensity of the signals, which correspond to the amount of the compound in the sample, over time, to see overall trends. Then they analyzed these curves for the rate constant and the reactions that were taking place. This is the creation of labeled GSH, where labeled cysteine is added to the cells. They then make the GSH out of the cysteine building blocks, resulting in a signal seen in the graphs. The signal is plotted over time on the right. This is a comparison between 1.2 and 30-minute NMR scans. And C shows the introduction of MBBR into the media, resulting in the reaction that I mentioned earlier. The addition of MBBR causes an increase in GSBIM and a decrease in GSH because the GSBIM is being created from it. After taking the scans, they plotted the intensities over time and fitted an exponential line to the data because the compounds will always follow these kinds of curves as they approach a set amount in solution. They saw that a 30-minute NMR was much more precise than the 1.2 minutes, but the fit on the curve for the 1.2-minute NMR was able to effectively capture the rate constant despite being less precise, which is just what they hypothesized that the NMR could do in the beginning. Thank you.